here. And today he's going to be talking about how he's really starting to embrace some of the uncertainty that we've been talking about in the past couple of days.
Thank you, Martin, for that very kind introduction. Good morning, everybody. It's really my pleasure to be here this morning. As Martin said, in our lab at Weill Cornell in New York City, we use both experiments on single isolated cardiomyocytes and computational modeling to try to study a number of problems, and we often integrate the two, the computational models typically running many of our experiments. The motivation for this particular area of work can really be seen by this figure here. This is action potentials recorded from about a dozen or so different induced pluripotent stem cell derived cardiac myocytes that are supposed to be all of the same type of ventricular cardiac myocytes. You can see that there's a ton of variability here. And so we're trying to understand this type of variability, as well as think about ways that we can better utilize this in the development of computational models. So I just want to first give you a bit of background on what the traditional paradigm is for developing computational models. Many of you know this very well. So you take a channel of interest, say a sodium channel, and express it in an expression system cell, say a human embryonic kidney cell. You then patch clamp it, deliver a series of voltage clamp traces. From those, you can record a series of current traces. The computational modeler then takes these and other current traces from the same channel type and develops a series of differential equations. These would represent just the differential equations for just the sodium channels, but as we know, a cardiac cell and models have many other channels and cellular components. There would be similar types of equations for all of these other channels and components. Then somebody, typically a graduate student, would take all of these series of differential equations and they would tweak them, most commonly with the maximal conductance of the particular channel, which is some representation of the number of channels in the cell, to obtain a particular output, typically a single action potential. So there are a number of problems with this particular approach. Some of them are described by this figure here that Steve published a decade ago for this particular human ventricular cell model, but this doesn't pick on this particular model. This same idea applies to basically all computational, cardiac computational models, at least that I'm aware of. What this shows are the origin of the data for the various channel equations. The letter indicates the species. Again, this is supposed to be a human model, but it has data from all these different species. The color indicates the temperature, which affects kinetics. So basically what you have are models built up from tons of data from different sources, also done in different labs with different protocols and different interpretation methods, perhaps. And so what you end up with are basically models of chimeras. So that's problematic. There are other problems, but I want to point out one more this morning. This is from Eve Marder's group. Eve is a neuroscientist, and what this shows are basically dynamics of bursting neurons. You can notice here that these three are basically qualitatively the same, and these are the two main driving conductances underlying these neurons. You can see that to achieve this same qualitative behavior, they have three very different sets of conductances. Then what they did was they took these three sets of conductances and they averaged them. And so this is the average model. You can think of this as the average model from three cells. But if you then simulate this average model, you actually don't get this behavior. You get a qualitatively different behavior. You get some bursting behavior. And so what this is meant to demonstrate is that, and this is just perhaps a bit of an extreme example, but nevertheless what it's meant to demonstrate is that when you take individual cells and you bulk them all together and average them, you might be missing some important underlying dynamics. With that in mind, a number of years ago we set out to basically ask this question. So instead of building a model from data from many different labs, cells, and conditions, can we instead construct models from intact cardiomyocytes? In that case, with all the components functioning, including subunits, so they're all present and they're interacting the way they're supposed to interact, and without ever averaging out the heterogeneity. Another way to think of this is that the protocols that I described previously are protocols that we as modelers are relying on from biophysicists who develop those protocols not for the purpose of modeling, but for their own other purposes. So can we actually start to think about developing protocols for developing models? And we think the answer is yes, and I hope I convinced you of that this morning. And so there's two main components of that. One is collecting information-rich data, and the other is that once you have that data, you harness the computational power that we all have in this era to tune all the model parameters in parallel. I'm going to spend most of the talk talking about the information-rich data because that's what we think is really the key, as opposed to the particular method of tuning the parameters, but I'm going to spend just one slide on this. So you saw this yesterday in Sam Wall's talk. So this is the method that we currently use. It's called a genetic algorithm, but we're method agnostic in this. There could be any number of methods that could accomplish this. So, but basically how it works, so this is a synthetic, basically it's a model fitting a model. And so this is a computational model output. The 9 parameters are maximal conductances for that model. The value of 1 for each of the 9 parameters are the normalized value that produced this action potential that we're trying to fit to. So we're trying to find the model that produced this action potential. So the way that these algorithms work is they take a bunch of guesses at these parameters. And so initially we take 500 guesses. And so here it shows 3 of them. This is a really bad guess because it has this red action potential or something like an action potential. This was a really good guess. At least in terms of the output, and this was something in between in terms of the output. So the genetic algorithm takes good ones and keeps them, throws out the bad ones over a series of 100 or whatever number of generations you use, and it ends up with a fit that looks like this. As you can see here, the fit model basically exactly or nearly exactly reproduces the target model, and it does so with these conductances. To my knowledge, this was first done by Ed Vigman's group about 15 years ago, and they used the approach more or less that I just showed to fit atrial data, experimental atrial data using an atrial model, and they achieved really good fits. But as you probably noticed, and as I think Sam mentioned yesterday, this fit is achieved by actually a model that's not the real model. Our goal here is to try to find the parameters of this real model because we're thinking about it applying it to a cell in the dish. We want to find the parameters of that cell in the dish. But to achieve this with something different than the real model. So how do we find our way out of that problem? This work by Eric Sobey's group kind of points to a path forward. And so this has the conductances on the x-axis as opposed to the y, but it's conceptually the same. So here's one set of conductances, here's another set of conductances. The same model structure, 2 different variants of that model. They both produce the exact same action potential. However, if you look at the calcium transient, the blue conductances have a different calcium transient than the red conductances. So now you can imagine that if you actually have only the action potential information, you don't know which of these is correct. If, however, you have the calcium data, you know, let's say, and it's blue, you know your algorithm will throw out the red one and keep the blue one. So the key here is that you need more information. Okay, so that's what we spend a lot of our time thinking about. How can we get more information from our single cell experiments to try to get closer to the actual model? So the first attempt that we made at this approach, and let me just throw out this, is that we work to develop protocols in silico, but we only consider protocols that we are confident that we can actually perform experimentally. We don't spend time thinking about, because we know we could come up with better protocols that might work in theory, but we only think about ones that we can do experimentally. So the first protocol, and Sam talked about this yesterday, has 2 parts. The first part of that is what we call aperiodic pacing. So these are pacing stimuli. This is in current clamp mode, and you record these action potentials. This is interesting because there's richer dynamics that are explored when you start to pace close together. So this explores different kinetic parameter space for the model. Then, in the same experiment, we then switch from current clamp mode, where you're recording voltage, to voltage clamp mode, where you're controlling voltage using this protocol and recording current. And so this protocol we worked on developing together with a biophysicist named Andy Zygmunt, who has really good intuition about the various channels. The concept here is we want each of these parts of this protocol to emphasize a particular channel to the extent that that's possible. So what does that mean? So this very first pulse here, what this says is this is the percentage of the whole cell current that comes through the various components, and they're listed over here. You may not be able to read it, but it doesn't really matter. So, but basically component number 6, which is IK1, 90% of the current is going through IK1 for this particular voltage pulse. So for your for a model that's developed to fit this particular part of the protocol, it has to have IK1 pretty close to correct, or it's not going to work. The same idea is throughout here. IK1 is the most identifiable in the term that Sam used yesterday. Some of them we really struggle with because of compensation between components, but that's the rationale. Okay, so if you do that, and this is still simulated data, this is what happens. So here are the parameters. These are the maximal conductances of the parameters. The nominal model that the algorithm is trying to find using that protocol has all the parameters set to 1. If you just train the genetic algorithm on a single action potential, what I showed a few slides ago, it doesn't find many of those parameters. So a genetic algorithm has some randomness built into it. And so you run it over, say 10 times. And so this shows the mean and standard error, the best guesses for the 10 runs. However, if you instead train it on the protocol I just showed one slide ago, which here is referred to as dynamic training, you have 11 seconds of data, it's able to find all the parameters really well. So this gave us some level of comfort and confidence that it was time to try to take this to the experiments. So the experiments that we performed at that time were on isolated guinea pig left ventricular myocytes. And so this shows, going into the experiment, the situation that we were facing. So here's a recording, a patch clamp recording of the red trace of 3 action potentials aperiodically paced. FR is a Luo-Rudy family model, which is the sort of leading guinea pig model. There are many different variants of it. They all basically have similar problems, is that they struggle, and it's not really meant to be a criticism of the model, they struggle to reproduce this type of data. So the Faber-Rudy model actually looks like this. So that was the situation going in. So in this particular experiment, which we published 4 years ago, we had data from 4 left ventricular guinea pig myocytes from 4 different animals. And these show the maximal conductances. And so the first thing that probably jumps out at you is like, whoa, that does not look like what you just showed me with the simulation, where it basically locked things down around 1 very tightly. But we don't actually expect that to happen, because we know going in that their model doesn't actually fit our data, so we know there's something wrong with the model. What we find encouraging about this particular study was that from 4 different cells from 4 different animals, there's actually clustering of several of the major components. I'll in particular point you out to the fact that these experiments suggest that IKR is 2 to 3 times higher in our cells than in the model, and IKS is much smaller in our cells than in the model. Also, back to this top figure here, the dashed line trace is the genetic algorithm fit, and so the model is able to really well reproduce the data. So we've since employed another approach for gathering information-rich data. It's called the dynamic clamp. Some of you may be familiar with this approach. The way the dynamic clamp works is you take a cell, in this case a guinea pig cell, you record data from that and it goes into your computer. And your computer is basically simulating a computational model. And for a particular voltage, it says, okay, at this particular voltage, how much current, let's say IK1, would be going through the membrane for this at this particular voltage. And so what you could do is you could take particular currents. You can say, you know what, if I want a little bit more IK1, I'm going to tell the amplifier, put in more IK1 into the cell. Okay, that might not have been totally clear, but I think when we get to the next slide, it hopefully will be. Okay, so these are experiments done by Francis Ortega, a very talented graduate student in the lab, together with Ryan Devenny. Ryan is a graduate student with Eric Sobey. And so what these experiments show are the black is a control case. It's one cell, a bunch of traces from one cell. And so the green is if the L-type calcium current is, say, increased by 40%. Through the dynamic clamp. The red is if IKR is decreased by 40%, etc. And so this is the recordings back to back in one particular cell. If you then simulate this experiment with a canonical model, you actually see it doesn't predict this very well at all, which is not surprising given what I just showed you. So there's a problem here. But what we could do is you can take these experimental data, and then you can put that back into a genetic algorithm and basically a long series of action potentials, and you can then try to train that without telling the genetic algorithm, like, hey, this green trace is meant to be a change in the L-type calcium. You just put it in sort of blindly, so you're not cheating. If you do that, a new model is adjusted, and this is the output of that model that very well fits the experimental data. This was done for something like a dozen cells, and each of these blue points are the mean conductances for those cells. What I'll point out to you here is that in these experiments, which were done sometime after the other experiments in different examples, we find more or less the same thing, where we have much less IKS, significantly more IKR, and there are some other similar changes as well. There's a fair amount of jumping around from animal to animal, so there's some heterogeneity, but there's consistency between this protocol, which is different than the protocol that we did previously. So perhaps they're exploring somewhat different parameter spaces, but they basically find more or less the same model. From a biological perspective, this is important because if your model has currents wrong, and say particularly IKR, if it has this balance of IKS to IKR wrong, its reliability for being used for predicting drug effects is really reduced. So more recently, we've been moving towards trying to get more high throughput by using human-induced pluripotent stem cell-derived cardiomyocytes. This is the slide that I showed on the opening, the figure on the opening slide. And we're doing these experiments for a number of reasons. One is that they are really a better model for studying heterogeneity and developing cell-specific model methods because they have so much heterogeneity, and you can do a lot more of these experiments more rapidly. Two is that they are becoming a widely used tool in safety pharmacology and drug screening, as you've heard in some of the other talks previously. And so we're interested in that as well. With regard to that second point, so here's action potential from an induced pluripotent stem cell-derived cardiomyocyte and from an adult cardiomyocyte, and they're very different, and you heard about this also. But basically, that's because these iPS cells are neonatal in their phenotype. They're not adult. And this chart shows some of the main differences, and in particular, we're focusing in currently on the fact that these cells have a lack of IK1, which is what causes them to be relatively depolarized at baseline, which is what also causes them to have basically some different plateau stage dynamics, which is critical for proarrhythmia, but also causes them to have a lot of spontaneous activity. So you saw the dynamic clamp before. You can use the dynamic clamp. We and some other groups as well have been using the dynamic clamp to try to get around that issue. In particular, if your model is simulating, you can focus just on, say, IK1. Let's say your iPS cell doesn't actually have IK1, but you can say if it did, let's inject a current that would represent that IK1. And if you do that, the blue is a control case of the cell under control mode, and the red is when you turn on the dynamic clamp. It hyperpolarizes the membrane potential to closer to what it should be. This is under pacing, so you don't actually see that, but if it's not under pacing but still under dynamic clamp, it also eliminates a spontaneous activity. Okay, so this is good, but that's what I just said here. However, I mentioned that we're trying to move towards being able to do things in more high throughput. And there's a big problem here that patch clamp is ultra low throughput. It's a very slow process. One cell, could be just a couple of cells a day or something like that. So a graduate student in the lab, Bonnie Quach, has been working on a project that we published last year in Biophysical Journal to try to replace, let's back up a step here, this red arm here, which is you compute the current, you inject it back through the electrode, to try to replace that instead with using an optogenetic approach. And so just very briefly, the way this works is that you express in the stem cells an opsin that's light activated and passes current as a function of the amount of light. Your computer says, okay, we need this much IK1, and you've done some calibration at the beginning of the experiment, and it controls the LED to titrate the amount of light hitting the cell in real time. And if you do that, the green is shown here, you can actually quite closely reproduce the dynamic clamp by injecting the current not through an electrode, but through the optogenetic channels. And so that's what's shown here. Bonnie did this in 16 cells, and she found that basically 11 of them did this quite well. So this is quite encouraging and promising, we think, because now you can start to imagine that this approach can actually be high throughput. You could have a bunch of cells in a number of different wells, each of them with an LED controlled by the computer, and with a different amount of light for each of these different wells. However, you also may have noticed a current technological limitation that we are facing, is that although the light is delivered, the current is delivered through the light, we're still using an electrode in these proof of concept experiments to measure the voltage. So we have to move away from that to a voltage indicator, so you can have both. But that's a technological limitation that we're starting to work on. All right, in the last couple of slides, let's just talk a little bit about other directions that we're moving in. As I mentioned at the outset, we're really spent a lot of time thinking about the richness of the information that we can gather. So the fit or the parameter identifiability will improve if we can increase the information content of the data. So here are three ideas that are, first, this one's fairly obvious based on the Sobey group slide I showed earlier. If we also gathered calcium data in addition to our action potential, that's a very important variable that would help us tune the model. You can also imagine that we can think about modifying the ionic concentration of the solutions, perhaps towards the end of an experiment. Relatedly, you can do very targeted drug blocks. Some of you may be familiar with Yi Chen Izu's work. It's called sequential dissection, where they add one drug to block a current, then they add another drug. You have to do it in a very well-designed way so that the sequencing is very key. But if there's a channel that our other techniques are not able to identify, we can think at the very end we may want to throw a drug on to block that channel, and that would improve the overall performance. And lastly, we're starting to think a lot about kinetics, and this is really inspired by the work of Kylie and Gary and the other members of their lab, who've had a number of nice studies and preprints that have started to think about not just tuning the conductances, which everything I've said so far is just about tuning the maximal conductances of the model. We haven't touched kinetics at all. But thinking instead about trying to develop protocols for also tuning the kinetics of the model. This is work from Francis Ortega in the lab, and this protocol, and this is very new stuff that we haven't published. It's very preliminary, but this protocol, for those of you familiar with their work, will recognize that there's a lot of similarities. Basically, you do a voltage clamp, and you also throw on like a 3-component sine wave, and this is meant to explore different kinetic space of the ion channels. So we think there's a lot of potential promise by going down this route as well. So in conclusion, I hope that I've helped to convince you that intact myocyte protocols can improve model fidelity and better illuminate cellular heterogeneity for a number of reasons. One is that the cellular components are all there. Any compensatory changes between components are all there, as opposed to if you're just studying channels in isolation. And you can do this by using information-rich protocols which you design specifically for this purpose as opposed to relying on data from biophysicists that was gathered for other purposes. And I mentioned, I think, pretty much all these, creating better protocols using these approaches, tuning model kinetic parameters, and we're also pushing forward on what I mentioned on those slides towards the end on using an optogenetic approach for the dynamic clamp, which will allow us to study more iPS-derived cardiomyocytes at a time. So thank you for your attention. So this is the group and the people that everybody who contributed to this. Trina, who's here, who many of you know, is involved in all of these projects and is very integral to everything that we do. Francis is a graduate student who worked on the dynamic clamp projects. Wilhelmine is a former postdoc who did the first study with the development of the dynamic protocol. And Bonnie is a graduate student who did the optogenetic work. And our collaborators, Annie Zygmunt, who helped us develop the initial protocol, Ryan and Eric on the project with Francis, and Amelia Encheva, who's helped us with the optogenetic work. So thank you very much, and I'd be happy to take any questions.
Okay, hi, thanks. Really cool.
Thanks.
There's something in the back of my mind, and I don't know if it's true or maybe outdated, but I always thought that in guinea pigs, IKS was much larger than IKR, whereas in humans it was reversed, and this is something you have to watch out for. And this kind of shows the opposite.
Yeah, we think that so there's a lot of changes depending on the adrenergic state of the cells, and we think that some of the earlier data I think that the cells may just have been in a different adrenergic state, but we've found this now in a couple of different studies, and I didn't show some data that we have where we tested this using a drug block after the fact that's really quite consistent with our data because this also concerned us. So we're not 100% sure why, but that's our leading theory as to why the cells in our lab seem to be quite different from some of the others. And we're not the only group, though, that has shown this, not in this way, but in other ways to suggest differences in level of IKS and IKR.
Do you think at some stage you might do this for every current, just to check if there's overlap, if the kinetics are in any way similar?
You mean the validation drug study after the fact? Yeah, well, what we'd really like to do, and I should have mentioned, is we'd really like to do some sort of single cell sequencing or analysis after the fact. We don't currently have the equipment or tools to be able to do that, to really validate this, because our validation is basically on that particular drug study after the fact, and the fact that the model reproduces the data, but it may be the wrong model. But we also base it on the fact that that in theory the approach works in silico, and so we and then two different protocols, which have actually quite different. So we think they're quite dynamically quite different. Also come to more or less the same answer.
David, excellent work. In your presentation, you never showed any restitution curve. Do you think it's still important to feed them, and do they reproduce?
This aperiodic pacing protocol that we use was inspired by the restitution curve, and so restitution curve, as you know, samples quite densely the dynamic space as you shorten the diastolic interval. But restitution protocols take a long time. And so we felt that not only were we not confident that you could, that reduces the number of successful experiments you can do. The longer the experiment takes, the more cells that are going to die during it. So we wanted to try to get at the same dynamics, but in a much more concise way, which is where this aperiodic pacing came from. And so, and that particular example I showed is not really the best example. Really, what we're moving more towards, oh wait, where is it? This type of protocol here is also similar to what I think Gary showed, which is many more frequencies or diastolic intervals. So restitution is key, but you don't necessarily have to use restitution to get at those dynamics.
Arlene?
Thank you, David. So I worked with GA a long time ago, and I remember that one of the difficulties we had is to find a cost function that gave you a good model, but also physiological complexity. So I'm just curious what kind of cost function you use.
So by cost function, are you referring to so basically what we let me just go back here. Sorry. The fitting function that we use is basically the error. So basically what we do is you deliver this same protocol, current clamp followed by voltage clamp, to your model cell, and you simulate and produce these outputs, this output in current clamp mode and this output in voltage clamp mode. So you basically have 11 seconds in this particular example of data. And then you do a mean squared error, point-wise mean squared error for all the data points throughout. There's different units here, and off the top of my head, I think we basically do some normalization, but that's the basic approach.
Oh, we should probably move on. Maybe we can ask one question while G-Chow gets.
Hi, so I was just curious about your data when you show that there's some spontaneous activity, and when you hyperpolarize it, it's gone. I was just wondering if you know why, the mechanism behind it.
Yeah, and so when it has to do with the kinetics and the recovery from inactivation of some of the ion channels. So basically, you need, when you're at this hyperpolarized state, there are ion channels that are basically opening and firing, whereas you move the hyperpolarized state, they're in a closed. They stay closed, and so they don't start you up the activation.
Thank you. Could you just make